Eritrichium is until now. And the objective of I've been working in this project for the last 4 weeks with Monty, Garut, and the Labicepi Ikarbon. It's pretty interesting. We learned how to distinguish between the three species of Alaska Eritrichium. And this project was introduced to the university by David Murray when he did his project on the Flora of North America project. He found little morphological differences between the three species. So we used phylogeny to get more genetics done to distinguish between them, and I will go through that later. And morphology is a study of the form and structure of an organism. So basically what color it is, how long it is, how many petals it has, and stuff like that, all the pretty stuff, you know. Molecular study was used with 3 chloroplast regions. The TRNSG was the gene that we used with the forward and reverse primers. And the only one that we got good results off of was RPS16 intron, which I will go over again. And the process that we used is we extracted the DNA and used the polymerase chain reaction, and we went through sequencing, which I will explain further. All right, you can see that these 3 species of Eritrichium, this is what we try to distinguish between. Eritrichium splendens over here, Iridioides, and Camisones. And basically, just by looking at them, you can't really tell the difference, but I can because I worked with it, and so can Monty and Stephanie. And so basically, more work needs to be done to distinguish the differences between them. All right, so basically you could see that there's a range map here, and then the green is Iridioides, which is found in the Seward Peninsula with the Camisones, and also in the Alaska Range with the splendens and Iridioides. And Camisones is also found in the Aleutian over here, and we got most of our samples from Kodiak National Wildlife Refuge. And there's a lot of Iridioides in the interior as well. And so basically these are the steps that we use to get our results. First we started out with the DNA extraction. We got preserved leaf tissue samples that were stored in silica gel, and we extracted that according to the DNA Easy Plant Mini Kit of the Kyogen kit. And then we went through a polymerase chain reaction, which was done to select and amplify the selections of the genes that we use, and once again the only one we got a good result out of was RPS16 gene. And the sequence analysis was the most fun. We got a lot of pictures out of it using the Cypress Science Gateway, maximum likelihood. We got really good results, and I'll show you that in a few more slides. Alright, so for the DNA extraction, we got a lot of Camisones up there. They're mostly from Kodiak. We got a couple from Baldy Village and one from Eisenbeck National Wildlife Refuge. And we got a few splendens as well from Bison Gulch and Donnelly Dome. And we got one sample of Iridioides from Eagle Summit. Basically, all the amount extracted, that's what we extracted from the tissue leaves. And they're basically in a good range, around the same amount. And then we did a PCR reaction, and we had used 4 programs using different temperatures and different timings. Pretty much the denaturation was between 94 and 95 degrees Celsius, and that is what separated the DNA. Then we went to primer annealing, which is from 51 degrees to 55 degrees, and that's just 45 seconds to a minute, and that is what the primer is attached to that I mentioned before. And then the elongation, which is set at 72 degrees, and that pretty much just makes it a longer strand for our results to come out conclusive. And then we hold it at 4 degrees until we use it to get the gel. And for this gel is made by gel electrophoresis in 80 volts for 45 minutes. And basically, wells 2 and 4 right here, that's what we want. We want a high concentrated product from it, and we got good results from Camisones and Iridioides, and you'll see that in the tree on the next slide. However, the splendens has a really low concentration. However, it's still good results for our data. And this is our tree that we came up with using cypress, and you can see that this is the only gene that we got results from, the RPS 16 intron up here. And then the Alaska Eritrichium are highlighted in blue, and the ones with the stars next to them are the ones that we did in our research. You could also see that the Camisonies and Radioides are closely related together and have an 83 maximum likelihood that they're related, and that's what we wanted. And then you can also see, however, that splenins are on the outside, so they're distantly related from the Camisonies and the Radioides in our study. And so basically, by just looking at these three species, you can't really tell like if they should be different species or the same species. If I was just looking at this on the first day, I'd be like, they look like the same species. Why do we need to do anything? But really, you gotta go in deeper and like get more results out of it. So basically, this is what we concluded from our tree, is that Eritrichium, Camisonies, and Radioides are very similar genetically, and their status as independent species may be put into question. And Eritrichium splenins appears distantly related to other Alaskan Eritrichium, and that suggests is that Eritrichium species have been introduced more than once to Alaska. And there are still very few highly supported ML bootstrap values in our phylogenetic trees, and more data must be required to do the two genes in our study that were not sequenced. And you can tell that in the tree that the splenins were outside and were distantly related. Maybe RAHI 2 next year we'll do another study on that using different genes to make it so, see if that's possible to make it so it's actually not part of other two species. I would like to thank my PI, Dr. Steffi Ickert-Bond, for letting me use her lab, and my mentor Monty Garut, and my lab partner Brittany Casperson, and the RAHI staff Denise Wartes, Greg Owens, Renee Pike, and the TCs except for Sean. RAHI 2 staff, Dr. Sue Hills and Amanda Meyer, and the RAHI co-instructors for helping me with my presentation, Dr. Kathy Katranis and Dr. Adrian Orr, and it was funded by UAF, Alaska BioPrep, Alaska Embry, and RAHI, and I'm pretty sure Sharon would have been bored this whole presentation. Any questions?